Hey, hello and good evening, everyone. So today we are in the week 8 problem solving session of the NPTEL course Molecular Biology. So I hope all of you have gone through the week 8 lecture video lectures. So in till week 7 we were dealing with the fundamental processes that take fundamental processes in molecular biology. Particularly, we looked into replication, transcription, translation, and uh, and we understood what are the how does the machinery operate in prokaryotes and eukaryotes. Now we are moving to understand what are the various techniques which are used in molecular biology. So understanding the concepts of these techniques are very important because if you join any lab, you have to regularly use these techniques. So you must understand the principle behind the techniques, and also um, how you can troubleshoot the various problems that arise when you are using these techniques for performing particular experiments related to your project. So some of you must have already used these techniques, or you have been using these techniques if you are working in any lab. So we will today we will understand the blotting techniques. So how we can detect protein, RNA, DNA, or a particular DNA, particular target RNA or target protein in a in a cell, right? Okay, so I will wait for 2 more minutes, and then I will start today's session. Okay, so I will start today's session. So let me share today's slides. Okay, so good evening, everyone. So I welcome you all to the week 8 problem-solving session of this NPTEL course, Molecular Biology. This course is being taken by Professor Vishal Trivedi of IIT Guwahati, and I am one of the PMRF TS for this course. I am Akash Jyoti Pathak, and I am PhD scholar in the Department of Biosciences and Bioengineering at IIT Bombay. So let's first quickly have a brief recap of what all we learned in week 8. In week 8, we learned mainly about the blotting techniques. Okay. So now we are moving on to the various techniques which are used in molecular biology. So one of the techniques which are used in molecular biology is blotting. So what is blotting? So there are basically four types of blotting as was discussed in the lectures also. One is the southern blotting. Southern blotting. Second is the northern blotting. Third is the western blotting. And another is the eastern blotting. So out of these four blotting techniques, these the first three are very commonly used. So southern blotting is used for detecting whether your DNA sequence has a or whether your DNA has a particular sequence. Uh, okay. Okay. Okay, so in week 8, we covered the blotting techniques. We learned about the blotting technique. So first of all, in southern blotting, what is southern blotting? Why is it done? So it is done in this experiment is carried out to identify or to detect particular sequences of particular nucleotide sequence in my DNA. So if you want to identify whether a particular sequence is present in the DNA, then this is the technique you have to use to identify a particular DNA sequence. Northern blotting is used for RNA, right? For identifying to analyze the abundance of a particular RNA transcript in a cell. Okay, so this is used for RNA. Western blotting is used for protein. So if you want to identify whether a particular protein is getting overexpressed or or what is the level of expression of of a protein in a particular cell, then you can use Western blotting. Or if you want to also if you want to understand whether there are certain post-translational modifications taking place in your protein, then also you can use Western blotting. Okay, for example, phosphorylation or ubiquitination, all these things you can understand using Western blotting. Okay, and Eastern blotting is used for identifying carbohydrates. Okay, so we will mainly understand the techniques, this Southern blotting, Northern blotting, and Western blotting. Okay, so first of all, we will understand Southern blotting. So Southern blotting is a technique which is used in so a practical example of where southern blotting is used is in forensic sciences. So if you want to identify, so you have a particular DNA sequence from a criminal site, and you want to identify uh, who is who is the particular so there are a couple of suspects and you want to identify who is the particular criminal among these suspects. So then yeah, from the sample any it can be hair or blood or from you can isolate the DNA and then you can do a southern blotting to match the DNA sequence of the sample with the DNA sample of the different So there can be 4 or 5 suspected criminals. So among the 4 or 5 suspected criminals, you can identify which is the particular criminal using this particular technique, southern blotting also. Okay, so let's understand the technique southern blotting. So what is done over here is first, so there are 4 to 5 basic steps in southern blotting. So first of all, for doing southern blotting, what do you need? You need to isolate the genomic DNA. Okay, you need to isolate the genomic DNA. So now the genomic DNA is very long, right? So what is done is this genomic DNA is then fragmented. You fragment the DNA, the genomic DNA, and how is that done? This is done by treating the genomic DNA with various restriction enzymes. Okay, so you treat the genomic DNA with restriction enzymes. So the restriction enzymes will digest at particular DNA sequences, and you will get short fragments of the DNA. Now, once you have the DNA fragment, the next step that is done is to run an agarose gel electrophoresis. So you you do an agarose gel electrophoresis, and you separate these fragments on a. So you separate the fragments on an agarose gel electrophoresis. Okay. Now, the fourth step. So once you have the different fragments, which are, so what will happen is the restriction enzymes will cleave the genomic DNA and it will create various DNA fragments. Now, these fragments, when you run on agarose gel, on an agarose gel, they will separate on the basis of their molecular weight or on the basis of their size. So you will get a pattern of DNA bands. Now, from these agarose gel, what is done? The DNA is transferred onto a nylon membrane or a PVDF membrane or a nitrocellulose membrane. Now, so why is this done? Because when you will use, so let's first, so why is then I will discuss later. So for what is done is that the DNA is transferred from the gel onto a membrane. Okay. Now in the next step, what is done is, so in the fourth step, what is the fourth step? Fourth step is transfer of DNA from gel to transfer of the DNA from the gel onto a membrane. Okay, onto a membrane. And the fifth step is you use a probe. You use a DNA. It can be a single-stranded DNA probe which is complementary to the target sequence. A probe is a DNA sequence which is complementary to your target sequence. You will probe against the target sequence. And then in the last step, you have to do autoradiography, or so if it is a radioactive probe, you will do autoradiography, or if it is any other fluorescently or enzyme conjugated probe, then you will use a particular substrate. So the enzyme will act on the substrate and it will produce a or it will produce a product which will give a particular color. Okay, so it can be a fluorogenic probe. Okay. So visualization. Last step is visualization. Okay. All these steps have been shown in this figure. So this figure is taken from the book uh, Benjamin Pierce genetics. This is a genetics book. As you can see here, so first what is done? The DNA. So here the first step is they are showing from DNA fragments, but actually what is done is so they have not shown the initial step. In this figure, the initial steps are not shown, but the initial steps are first you have to isolate the genomic DNA, you have to fragment it by various restriction enzymes. Now, so once you get the fragments, then these fragments are separated by gel electrophoresis. Then from the gel, you have to transfer the DNA onto a nylon membrane or a nitrocellulose membrane. Why? Why is this step required? Why can't you directly add the probe on the gel? Why can't you directly add the DNA probe on the gel? So this is necessary because the if you use if you, for example, you are using a single stranded DNA probe against your target sequence, this probe will not be cannot enter the gel. So you have to transfer the DNA from the gel onto a nitrocellulose membrane. And how is this done? So this is done by the principle of capillary action. Okay. So what happens is as you can see in this figure, there is a support, and on the support, the blotting papers are placed. So what will happen is blotting papers will the so this blotting paper is placed such that they can dip in the buffer. They these are dipped in the buffer, and so by the process of capillary action, this buffer will move through the blotting paper, and then the water will be soaked from the as the water gets soaked from the blotting paper onto the gel, the DNA will move from the gel onto the nitrocellulose membrane. Okay, so buffer drawn up into the top layer of the blotting paper, this buffer passes through the gel. So this buffer will start passing through the gel, and along with it, what will it carry? It will carry DNA onto the membrane. Okay, so you can see here in this figure, so this is your gel, and after this blotting process, the DNA has been transferred from the blotting paper onto this membrane. Okay, so you get a the pattern that was there on the gel, you get a exact pattern on the blotting paper or on the sorry on this membrane, on this nylon membrane. And now what is done is now you have to once the DNA is transferred onto the membrane, you have to add the probe. You have to add the it can be a labeled probe or a radioactive probe, which is complementary to your target sequence. So so what is done? So DNA on the membrane is fixed. So before this, what is done is that the DNA is fixed on the membrane by placing it in a ultraviolet chamber or a high ultraviolet light is passed, and the DNA gets fixed on the membrane, and then this membrane is placed in a hybridization bottle with solution that contains radioactively labeled probe and gently rotated. And after this step, so what will happen? The probe will bind to particular sequences or sequences that are complementary to the probe sequence. It will bind to those regions, and then you can do autoradiography to detect fragments with probe attached. Okay.
Sir,
So yes.
so what you said about that reason, capillary action, that means what was the
Hmm.
reason for? Yes, sir.
What was the reason? Sorry, can you repeat?
So what was the reason for not having probe with the gel?
So your gel is very thick, right? So have you prepared agarose gel? Have you seen an agarose gel in your lab or?
I
Uh,
have seen but not prepared yet.
okay. Okay. So what happens is the gel that you prepare in a lab, that is very thick. Okay. So,
Mm.
so So the gel is very thick. So what you have to do is now if you add the probes, if you add the probe and the DNA sample will so what you do is you load the sample on the well, and then you apply the current. The DNA will start moving from the towards the positive electrode, right? Now the if you add the probes, if you add the probe, then because the gel is very thick, this probe cannot penetrate the gel and bind to the DNA. So what is done is by the through the process of capillary action, the buffer. the buffer is the this buffer gets soaked by the blotting paper, and from the blotting paper, this buffer starts moving upward, and as it moves upward, the DNA is transferred from the gel to the membrane. Now this membrane is thin, so the probe can directly attach to the DNA which is present on your membrane. Okay, did you understand?
So, but probe is of smaller size, then why it can't go in that thick membrane? DNA is going, but why not probe?
So what happens is when your DNA is moving through the gel, the DNA is present inside the gel, right? It you have to so how will this probe enter the enter inside the gel?
So,
Uh,
when if we will put in the well with the DNA
mhm,
itself, then it can enter the gel.
mhm. So you are
So,
applying an external current so that this DNA can move through the gel, right? Through the pores which
hmm,
are present on the agarose gel, in the agarose gel. So you have to apply an external, uh, so you are applying an electric field so that it can, this DNA can move through the gel. But on its own, so if you just load the sample on the into the well, then this DNA cannot move on its own. You have to apply an electric current, right? So
hmm.
for that reason, if you just add, so if you take the gel and then you dip it in your buffer and you add the probe, how do you expect it to um, move inside the gel and bind to the DNA? So you have to either apply an electric current. So there are electroblots that we will see in Western blotting. So in Western blotting, you apply a current so that the protein can move upward. So I will discuss uh, when I will talk about Western blotting. So you have to, so here you have to, this probe cannot directly enter the gel. Okay, that is not possible. Is it clear now?
Okay, sir. Once Western
Mm.
blotting is completed, then maybe I can understand
Yes.
more.
Either you have to apply an electric current so that the DNA can move. Now that the DNA has separated on the gel, you can again apply a current so that this DNA can move from bottom to the top, or you can attach a positive electrode over here and a negative electrode over here. And then you, once you close the chamber, this DNA can be transferred to the membrane. That is possible. Or otherwise, this another method is by doing capillary, by the capillary action, this DNA gets directly transferred onto the nitrocellulose membrane.
So, sir,
So
means with
mhm.
the probe, it can't be transferred. Means if we put DNA along with probe in the same well, then it can't be transferred. Means we can't get the band.
So, so why do you are telling that you will load the probe along with the DNA?
Yes, sir.
So, so in that case, if you have many samples, you have to load that
Sir, is the probe is the probe interact will the probe interact with the gel, or will the probe interact with the DNA? It will interact with DNA only.
yeah, it will interact with DNA, but but in that case, the probe will be will but actually, why do you want to separate the probe? Like the probe will also get, if you apply electric current, then the probe will also get separated along with the DNA. So so that method is not actually done. Okay. So you have to add probe to all the wells, and then you are also these probes are actually costly. So if you add probes to all the samples and then you run the gel, then it will be expensive also. Okay. Okay. So the southern blotting, what is the purpose of this technique? The purpose is to verify sequence identity of the sample DNA fragment. So you have a particular, you're interested in a particular gene sequence or a particular gene, and you want to identify whether this particular DNA sequence is present in the cells of in a particular cell. So for identifying the sequence, you can use southern blotting, and the DNA or RNA probe is which is used is complementary to the target sequence of interest. And for visualization, what is done? The probe is labeled with radioactive molecule or fluorogenic molecule. Okay, and also, so this is also one reason. So your probe is basically labeled with fluorogenic molecule. Okay, so if this is your probe, it will be labeled to a particular fluorogenic molecule, and then once you add the substrate, it will convert it into a particular product. Okay, or when you shine light on it, or if you excite it with a particular radiation of light, and it will emit a particular radiation, and then you can analyze it. Okay. Now there are six main steps as I discussed. So first is genomic DNA isolation, then digestion of genomic DNA, then agarose gel electrophoresis to separate digested fragment, transfer of DNA from gel to nitrocellulose membrane, hybridization with radio-labeled probe, washing and autoradiogram. Okay.
Sir, what is the composition of nitrocellulose membrane?
So nitrocellulose membrane, it has uh, it is actually a polymer of so cellulose and nitro. So what is the composition, exact composition? I also have not checked. Nitrocellulose means it will be a polymer of.
What is the composition so that it can interact with DNA?
So it should be positively charged because your DNA is negatively charged, and so that it can interact with the DNA. And also this nitrocellulose membrane also you have to activate the membrane. You have to keep it in a particular buffer so that it gets activated and then can bind to the DNA. Okay. So the genomic DNA is digested with one or more restriction enzymes, and the fragments are separated by agarose gel electrophoresis according to size. And then the DNA denatured. Another thing what is done over here is that you need to denature the DNA, right? Because if you want your DNA to attach to a particular probe, to attach to a particular DNA or RNA probe, you have to denature the DNA. So before transferring it to the nylon membrane, the DNA is denatured. Okay. So as you were saying that if you add the probe directly to the DNA, and then we load the sample into the gel and run the agarose gel electrophoresis. So in that case also, so the DNA is double stranded. So how will you So you have to either this will also be not this won't be possible, right? You have to denature the DNA, you have to separate the two strands, and then you can add a then you can add the probe. Okay. If you denature the DNA and then run the agarose gel, then uh, the particular molecular weight of the DNA that will also be changed. Right. So you will not be able to identify that. So you are getting, so you run a ladder, right? Run a DNA ladder along with your sample. So if you add the probe and then you load the sample, then you will not be able to understand what is the size of the fragment, or what is the sequence to which my probe is binding. So there will be many problems. That is actually not cannot be done. Okay, so the DNA denatured and transferred to nylon membrane or nitrocellulose, and DNA attached to membrane are hybridized to label DNA, RNA, or oligonucleotide probe, and the bands complemented to the probe are located by autoradiography. So this is how the setup of a southern blotting looks like. So you have a support, and on the top of the support you add a Whatman paper or a blotting paper. Okay. So now what will happen? This Whatman paper will get stored by this transfer buffer. So this transfer buffer, there are particular composition. This was explained in your lecture slide. What is the composition of the transfer buffer? For how long you have to this process is required. What all optimization is required. So I'm not going into those details. Um, so there are specific protocols that you have to follow. So then also on top of this Whatman paper, you place your gel, agarose gel, and then on top of the gel, you place your nitrocellulose or nylon membrane. Okay. Now on top of this nitrocellulose or nylon membrane, you add the Whatman. Again, you add the blotting paper to absorb the excess water. Okay, absorb the excess water, and then you add some weights. You add some weights so that uh, so that by putting on weights, you can enhance the transfer of the DNA from the gel onto this membrane. Okay, so the DNA fragments are carried from the gel by capillary action. And this is the capillary action. How will you maintain this capillary action? By adding a stack of dry absorbent paper towels. So these are paper towels. So to maintain the capillary action, you add a couple of towels, and then the DNA is eluted from the gel by a moving stream of buffer. Um, and as the buffer moves, what will happen? The DNA will get deposited on the nitrocellulose membrane. Now answer this question. So there are six major steps of southern blotting techniques, as I discussed. So here also they have been labeled as A, B, C, D, E, and F. Okay, so here what is A is DNA isolation. B is what? B is digestion of DNA. C is transfer of DNA from the gel to nitrocellulose membrane.
Option A.
Option A. Okay. By option A. So the first step is DNA isolation, right? That is
Yes.
correct. You have to isolate the genomic DNA. Then the second step is digestion of DNA step. This is also in sequence. Then, then transfer of DNA from gel to nitrocellulose membrane.
No, no.
So this
Then
is no.
agarose electro.
Hmm. Yes, first you have to separate the DNA fragments by running an agarose gel electrophoresis. So yes, E, first E, then C, then radio labeled probe attachment, and F is washing and auto radiogram. So why you need to do the washing step? Why you
To
need to
remove
do
the unbound.
or the non-specific by non-specific binding of the probe to the DNA. Okay. So for example, if you have a particular sequence, for example, AAG, C, CA. So this sequence can be how many times can this sequence be present in your genome? Can you calculate? So if you have a genome, for example, your genome is of mm, some 100 kb or 200 kb genome. So this can be present multiple times. So this sequence can be present multiple times in your DNA. Okay. So there is a possibility of non-specific binding. Also, non-specific binding means, for example, your target, your probe has the particular sequence GG, TT, CCAT. So now this sequence will also bind to this part, GGTT. But this binding will be of a lower affinity. So when you wash it, when you wash the this your membrane, these non-specific binding can be removed. Okay, so these are also problems. So if you what you were saying, asking me that if you directly mix the probe with your DNA. So for example, you have denatured your DNA. You have denatured the DNA, and then you have added the probe. So there will be many non-specific binding. Okay, and how will you remove the non-specific binding? So your gel is thick. You cannot wash it. So your membrane, you can wash your membrane for removing the non-specific binding. But if your DNA is trapped in the gel, how will you wash it for removing the non-specific binding? So that is also not possible. Okay, and you will see multiple that there will be multiple DNA sequences to which your probe will bind. Okay, so that is also another issue.
So this in the case AAG, this one is probe and the other one is the strand.
No, so this is your target DNA sequence. So this is the particular DNA sequence you are interested in. So, for example, this is your gene sequence. So, for example, this is the gene which codes for hemoglobin. Say, for example, just for an example, say this is the particular sequence which is coding for a particular protein hemoglobin, and you want to identify whether this sequence is present in a particular say for example any on any cell of any organism, you want to see whether this cell can produce this protein. So this is your DNA target DNA sequence. This is your target DNA sequence. Okay. And this is this GGTTCCAT. This is the probe. This is the particular probe that you have used for. This is the probe that you have used for identifying whether whether your DNA sequence has this particular sequence of nucleotides. Did you understand?
So not getting
You are not
clear.
getting. You are not, okay. So you get you you have fragmented your DNA. Okay. Now after fragmentation of the DNA, you are running it on an agarose gel. Okay. Now before transferring the DNA onto the nitrocellulose membrane, you denature the DNA. So how do you denature the DNA? What you do is you add NOH to the solution. So NOH will what it will do, it will NOH and NS salt concentration. This NOH and salt concentration will separate the two strands of DNA. So now from double-stranded DNA, you will get single-stranded DNA. Okay. So now this single-stranded DNA will be transferred from the gel onto the nitrocellulose membrane. So your nitrocellulose membrane will have single-stranded DNA bands. Okay. So this is your, so there will be multiple DNA bands, and this is the particular DNA sequence you want to identify whether this particular sequence is present in your genome. Okay, so this is your target sequence, and for identifying so this is not actually the target sequence. So this is another this is not the target sequence. This is any other sequence which is present. Okay, so the target sequence that you want to identify will be complementary to the probe sequence. So this is any other sequence which is present in your DNA. So if you have a probe sequence, if you have a sequence have a DNA probe whose sequence is like this GGTTCCAT, then this probe will also bind to this DNA sequence, and it will also bind to the DNA sequence which is having this sequence as well, right? So for removing this non-specific binding, you need to do the washing step. Okay.
So sir, means the probe will bind with the target sequence, or it will bind with the DNA fragment. It will bind with the DNA fragmented
It will
sequences.
bind to the target sequence only because the target sequence will be exactly complementary
Probe.
uh, to your probe. But at the same time, there are chances that it can also show non-specific binding, as I was giving you this example.
So means
So, for
the
example,
fragmented which is not complementary to that probe will show
they are not
non-specific.
exact. Yes, they are not exactly complementary, but some regions of the probe sequence might be complementary to any other sequence which is present on your membrane. So in that case, it will show non-specific binding, isn't it?
Got it, sir.
Is it? Yes. Is it clear now? So your target sequence, for example, your target sequence is of how many base pair? How many bases? One, two, three, four, five, six, seven, eight. So out of these eight bases, if there is any other sequence whose four bases match with the four bases of the probe, then it will also go and bind to that sequence of DNA. So for that reason, you need to do the washing step. Okay. And so for that reason also, what you were asking initially, that if we directly add the probe to the DNA, then can we not see the target, where can we not identify the target sequence? So that will be very non-specific. You will get many non-specific bands, and you will not be able to identify which is your which is the correct correct signal. Okay, for these reasons. Okay, so this is directly from the, I've added the slides from the video lectures. So here also this was nicely explained. So there are 6 steps: genomic DNA isolation. Then you digest it to generate fragments. Then you separate it on an agarose gel, and then transfer of DNA from gel to nitrocellulose membrane. Then hybridization followed by washing and autointegration. Okay, now answer the next question. During the process of genomic DNA isolation, disrupted cells are incubated with a digestion buffer containing dash to release genomic DNA from DNA protein complex. So when you want to isolate the genomic DNA,
protease
um,
and
yes, protease and SDS. So can you explain the answer? Why do you need to add protease and SDS?
protease to degrade the proteins because it
Hmm.
is a DNA protein complex, so DNA will get separated.
Mm-hmm. Yes, absolutely. Yes, correct. You are correct. So the DNA is bound to your histone proteins, right? As we had discussed in this nucleosome, the genome organization, when we were discussing that the DNA remains bound to histone proteins. So to remove these, so if you want to isolate the DNA, you need to you need to add protease so that these histone proteins and other proteins which are present in the cell lysate, they can be digested as well. Okay. And any if there are membrane protein, then this SDS can also act on these. Okay. So the SDS also denatures the protein. SDS, protease, all these will denature the protein into peptide, and um, you can get your genomic DNA. Now answer this question: purification of genomic DNA with dash. So this was also discussed initially also in lecture 2 or 3, isolation of DNA, isolation of RNA, these were discussed. So purification of genomic DNA with dash mixture has 2 phases: aqueous phase and organic phase. In this step, phenol denatures the remaining protein and keeps the protein in the organic phase, and genomic DNA is present in
Phenol
the,
chloroform.
sorry,
Phenol chloroform.
phenol chloroform. So what happens when you treat your genomic DNA with phenol chloroform?
So what was the ratio? 24:7.
Yes. Ratio is, yeah, it is 24:7. Phenol chloroform, and sometimes isoamyl alcohol is also used. 24:7:1.
One is
In
to
that
one.
ratio, it is used. Isoamyl alcohol is used. So what it does is this: the proteins and the lipids, they are soluble. They are soluble in this organic phase. So your protein and lipids, they are present in the organic phase, while the DNA remains in the aqueous phase. Okay. So the answer is phenol chloroform. So yes, so when you add phenol chloroform mixture, the chloroform, this mixture will have 2 phases: aqueous phase and organic phase. In the organic phase, what will be present? Your lipids and proteins. Okay, lipids and proteins are present, and in the aqueous phase, the DNA is present. So what you do in the next step? So you spin it. You spin it, or you centrifuge it, and then you isolate the aqueous phase. Then you again, what you do is, because now this aqueous phase will have both chloroform and phenol. Okay. You do not want your DNA to be contaminated with phenol chloroform. So if you take a reading of a DNA having phenol, then it will give a very high reading, because phenol also absorbs at 260 nanometer. So what is done is that the once you isolate the aqueous phase, you again ethanol precipitate it. You precipitate it by using 100% ethanol. So when you add 100% ethanol to this aqueous phase, the DNA will get precipitated, and then you can isolate the precipitate and dissolve it in your required buffer. It can be any tris NaCl buffer or any required buffer that you want to work on. Okay. Okay, so now let's learn about northern blotting. So northern blotting is, so all these blotting techniques, they are somewhat similar. Okay, why is it similar? In some sense, you need to run a gel and then you have to transfer your sample from the gel onto a particular membrane. So that step is common in all the three blotting techniques. Okay, so now what northern blotting is done to identify what? To identify your mRNA sequence, or you want to identify a particular particular RNA transcript in your uh, in a population of RNA. So, for example, you want to analyze the expression of a gene. Okay, so if you want to analyze the expression of a gene for example, you want to analyze the expression of a gene in two different cells, in a cancer cell and a normal cell. So, how will you do that? So, for example, there is a particular gene, and you know that this particular gene is expressed in a particular cancer. So, you want to understand what is the level of this expression, or for example, you have treated this cell with a particular drug, and then you want to check whether the expression of this gene is going down, or whether it is getting upregulated, downregulated. So, such studies you can do by carrying out northern blotting. Okay, so now now when you isolate the RNA from the cell, there will be many types of RNA. There will be 5S RNA, 5.8S RNA, there will be tRNA, there will be rRNA. Okay, but one feature of the mRNA that differentiates it from other RNA is that it has a poly A tail. Right, as we had discussed that the mRNA will have a poly A tail, and now using you can exploit um, this the distinguishing feature of the mRNA to isolate it from the other RNA which are present in your sample. So what you do is that since the eukaryotic mRNA have this poly A tail, what you can do is you will use a column. You will use a column. Now these column have resins. Okay, so these columns have resins, and these resins are so these resins are primed with poly T tail. So they will have a oligonucleotide attached to this cellulose beads. Okay, so now when you add the RNA, add your isolated RNA through the column, only the mRNA will bind to the mRNA will have poly A tail, and they will bind to this beads which are tagged with this poly T sequences, oligo DT sequences, and then you can. So once the binding is complete, and then you can elute it by changing the buffer conditions. Okay. So what is done is that the mRNA are separated by affinity chromatography on oligo DT cellulose columns. Okay. So the poly A tail has the ability to form stable DNA RNA hybrids with short chains of oligo DT linked to a supporting cellulose matrix. So as I was explaining that there are particular columns, and these columns are have these resins. So these resins are these resins have a are having this oligo DT oligo DT nucleotide sequences, which are around 18 to 30 nucleotides in length, so that when you pass the mRNA, they will bind to this poly they will bind to this column or to these resins. And then what is done is high salt is added initially to stabilize the nucleic acid duplexes. Okay, so why do you need to add high salt? Is because there will be high repulsion because of the charge between the negative charge, they will repel. So first of all, high salt is added so that this repulsion can be electrostatic repulsion can be removed, and the mRNA molecules can bind to this reasons having this oligo DT nucleotide sequences, and then low salt buffer is used to destabilize the double-stranded structure and elute the poly-A RNA. Okay. So the northern blotting is used to measure the amount and size of RNAs transcribed from the eukaryotic gene and to estimate their abundance. Okay. So you can also estimate the abundance. So as I was explaining that if you want to understand the expression, so what is the how in what amount that RNA is getting expressed or being expressed by that cell, that also can be understood by northern blotting. So here also, what are the basic steps? You will isolate the intact mRNA. Then, so before this, first you will isolate the RNA from the cell or the tissue. And then from the RNA, you need to do affinity chromatography using oligo DT column. And you will isolate the pure mRNA. Okay. And now when you have the mRNA, you have to separate the mRNA similar to what you were doing for DNA in southern blotting. So you separate the RNA according to size through a denaturing agarose gel. So this is one thing you have to note over here is that for RNA you use a denaturing agarose gel. And why is it so? Why do you require a denaturing agarose gel for RNA? So this is because this RNA with that single
To
stranded, like
separate
RNA.
to
Hmm.
separate the RNA fragments.
No, not the RNA primers. The RNA are single stranded, so
RNA
they can
fragments,
form.
sir. RNA fragments, not primers.
Yes. Okay, okay, yeah. RNA fragments. So these RNA fragments can assume any secondary or tertiary structure. Okay. So to remove these secondary structure, you need to prepare a denaturing agarose gel, and this denaturing agarose gel is prepared by dissolving this agarose gel in formaldehyde, in 37% formaldehyde. So what will happen is all these single-stranded RNA which are having different conformations, they will get denatured, they will denature and they will form a linear single-stranded RNA structure. Okay. So then the next step is you transfer the RNA to a solid support, then fix the RNA to the solid matrix. Then hybridize the immobilized RNA to probes complementary to the sequence of interest. And then the removal of probes that are non-specifically bound to solid matrix. And the last step will be detection, capture, and analysis of an image of the specifically bound RNA to the solid matrix. So these are the basic steps.
So solid support and solid matrix are the same.
Non-specific, yes, yes, yes. Solid matrix and what are you asking? Solid
Solid
support,
support.
yes. Yes, 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 yes. They're the same. Solid support and solid matrix.
Okay, sir.
So here we have to note that you have to run a denaturing agarose gel, and this denaturing agarose gel is prepared by adding 37% formaldehyde to the RNase-free water. So working with RNA is difficult than working with DNA because RNA gets degraded very easily, and RNase is present everywhere. It is present in our sweat and yeah, so you have to be very careful when working with RNAs. You cannot talk, you cannot work with free hand. You have to wear gloves and work. So, and also the water that you're using, that should also be RNase free so that this RNA does not get degraded. And here also nylon membranes are used for the transfer of RNA from agarose gel. And why are nylon membranes used? Because they're more durable. They are more durable than nitrocellulose filters. Okay, and nucleic acids can be immobilized on nylons in buffers of low ionic strength. So transfer of nucleic acid from gels to nylons can be carried electrophoretically. Okay. So so during the fixation step, as I had discussed here, no, the fixation of RNA to solid matrix. So here either you have to apply heat, yeah, you have to bake the membrane at 80 degrees Celsius. So when you heat it, the nitros these nitrocellulose filters, they are not very stable, and uh, they assume different or they are not stable at such high temperatures. So nylon membranes are better in these cases. Okay. So during the process of northern blotting, so I'm not discussing each and every step of the northern blotting technique. So those were explained in your lecture slides. So what happens? So I'm going to the question. So during the process of northern blotting technique, which component is used as a blocking agent? So why do you need a blocking agent? So what happens is when you once you have transferred the RNA from the gel onto a nitrocellulose membrane or a nylon membrane, you have transferred the RNA, you have transferred the RNA from the gel onto a nylon membrane. Then before adding the RNA probe, complementary RNA probe, you need to add a blocking agent. So you need to dip the membrane in a buffer which is having skim milk or BSA. Okay, so that there is so what happens so that there is no non-specific binding of the probe to your membrane. So you want your probe to attach to only the particular target DNA, particular target RNA sequence. You do not want the probe to attach anywhere on the membrane. So if what will happen if the probe attaches anywhere on the membrane, then once you visualize it, or when you add the particular substrate, or if it is a fluorescent probe, then you will get signals everywhere on the membrane. You want the signal at only a particular from only a particular uh, at only a particular RNA sequence. Okay, so for that reason, you need the blocking agent, and the answer is Salmon sperm DNA is used in case of northern blotting. So in case of this northern blotting, the this particular salmon sperm DNA is used. Okay, so this DNA, it will bind to different to different regions of the or to the regions of the membrane which are free, and once you add the probe can will only bind to to your target sequence, and it will not bind to the other regions of the membrane. Okay. So this is actually wrong. So this BSA casein or non-fat milk, this is actually used for Western blotting, not Northern blotting. For Northern blotting, you need salmon sperm DNA as the blocking agent. Okay. Now, which feature of mRNA is mainly responsible for isolating the mRNA population from the RNA pool using a poly-T affinity column chromatography? So this I discussed just now.
Poly A tail.
Yes, poly A tail. Okay. And why is it so?
Because poly T will only bind to poly A tail.
Yes, poly A tail, because the mRNA has poly A tail. So the mRNA will go and bind to this to the column having poly T affinity, having the poly T resins. Okay. So yeah, so if you see the structure of mRNA, you have a cap, 5 prime cap, 7 methyl guanosine residues. Then there is a 5 prime untranslated region. Then coding sequence, 3 prime untranslated region, and then there is poly A tail, which are composed of around 200 residues. Okay, so this feature is utilized for isolating the mRNA from the other population of RNA. So yes, so yeah, the nucleotide adenine forms two hydrogen bonding with nucleotide thymine, and this pairing is very specific. Exploiting this feature, mRNA population can be isolated from RNA pool using a poly-T affinity column. Okay. So what is the primary purpose of novel blotting? This question is very easy.
RNA RNA fragment detection.
RNA fragment detection. So this does not require any explanation. So now answer this question. Why are RNA gels performed under denaturing conditions? So you read the question and the options nicely and then answer what will be the correct option.
Option A.
Option A. Why?
Secondary structure, if it will get
Hmm.
migrated faster, then
Hmm.
it will means the for the molecule it will take less time to interact with it,
Agarose
as
gel, yes.
speed is faster. And as you
Hmm.
said that RNA forms secondary structure, so to
Hmm.
denature that structure we need what we need
We need to prepare a denaturing gel.
that formaldehyde
Hmm.
we need that. So
You need to mix formaldehyde with the agarose and then cast the gel. Okay. Yes, so they are performed under denaturing conditions. Yes, correct answer is option A. So the RNA gels are performed under denaturing conditions. And why is it so? Because of the presence of secondary structure in RNA. Now, if secondary structures are present in RNA, then what will happen? It will migrate faster. So as you had seen in DNA also. So if you run a plasmid through an agarose gel, then the plasmid has three conformations, right? What are the three conformations that are present in a plasmid? The supercoiled, open circular nicked, and the
so
circular DNA. Right. So the supercoiled form of the plasmid migrates faster. So you can see three bands, right? If you run a plasmid in an agarose gel, you will see three bands. So the band which migrates the fastest is the supercoiled DNA. Then the open nicked DNA, linear DNA, and then on top you get the circular DNA. So similar here also in case of RNA, because RNA has different secondary structure. These secondaries RNAs having secondary structure will migrate much faster on agarose gel. So what are the different secondary structures? They are stem, hairpin loop, as you had seen in tRNA, structure of tRNA. There were different, they assume different 3D structure, tertiary, secondary structure, because of the complementary regions. They can form hairpin loop, pseudo knot, bulge, internal loop, multi-loop. So all these, what it does is it gives less time for the molecule to interact with the agarose gel and consequently less resolution within the different RNA species. So destruction of the secondary structure in the RNA structure minimizes these effects and allow better separation on agarose gel. Okay. So these are easy questions, right? So the techniques are easier compared to the transcription translational machinery, because there you have to remember the name of many proteins, transcription factors, initiator protein. So the here you can, if you understand the concept, you can easily solve these questions. Now answer this question during the Gober-Hoffman method approach for cDNA synthesis. After first strand synthesis using oligo-DT primer in the presence of DASH and DNA and DNTPs, the DNA-RNA hybrid is treated with DASH to produce nicks at multiple sites. Then DASH is used to perform DNA synthesis using multiple fragments of RNA as primer to synthesis new DNA strand.
option no option A, reverse transcription; B, restriction enzyme; C, DNA polymerase.
This one, oh, actually, this I have not labeled. This is A, B, C, D. Which option you are telling? Option
Option
B.
B. Yes, sir.
Option B. No, option B is not the correct answer. So how do you synthesize cDNA? So do you know what is complementary DNA?
Yes, sir. Oh, the
Yeah.
strand which is complementary to means
No, no, that is not complementary DNA. So if you synthesize, so you have an RNA.
so for
You have a
that we
RNA.
use an mRNA
Yes, you
and
are
then
correct.
synthesize from that strand that is called CDN.
Yes. So cDNA, what is the full form of cDNA? So if you synthesize a DNA from mRNA, then that is called complementary DNA. If you synthesize the DNA, how do you synthesize DNA from mRNA? Why is it called complementary DNA? Why is cDNA called complementary DNA? And why is complementary DNA synthesized?
Because with that mRNA we first means w- one more strand is added and then we cut the first strand with
You are correct. Actually, you are correct. But why do you why is cDNA synthesized? Or what is the advantage of synthesizing cDNA? Uh, or you already have a DNA sequence, right? You already have a DNA sequence. Why do you need to synthesize a DNA sequence from an mRNA? Have you ever thought why the cDNA? There are different cDNA libraries. So we will in the later lectures, these will also be taught cDNA libraries. Why are they required? So why, what is the main reason or one of the fundamental reason? So basically, if you see a eukaryotic DNA, then your eukaryotic DNA will have both exons and introns, right? So there will be regions within the DNA which do not code for any protein, and you would not want that region to be present. You want to have only that DNA sequence. Which is not having introns. So if you want a DNA sequence which is not having any introns, then what you can do is you can using the reverse transcriptase enzyme. Using the reverse transcriptase enzyme, you can synthesize a DNA sequence from the mRNA. Now this complementary DNA will not have any. It will not have any introns in it. So now you can directly use this complementary DNA. You can insert it in a plasmid and then you can expression of a protein. Okay. Did you get it? If you directly take, so for example, you want to, you will learn about this recombinant expression of proteins and all these in later lectures. But because the concept of cDNA came, I was just giving a fundamental idea of what are complementary DNA. So basically, if you want to express any, for example, you want to do recombinant expression of insulin. Now this insulin uh, gene, if you look at the insulin gene in a eukaryotic genome, this insulin gene will also have introns within the exons. So, but now if you want to recombinantly express this insulin in a bacterial cell, you will not want the introns you would not want the introns to get cloned in your plasmid. Okay, so for that reason, what you can do is you can reverse transcribe the mRNA into complementary DNA and then you can clone it in a plasmid so that you can express the protein and then further whatever
so sir we can
and do your
use that with the help of mRNA also. Why we are taking
what
means
is what is your question? We can directly use
mRNA.
mRNA. How will you? how will you mRNA? Can you transform a bacterial cell with mRNA directly? If you
Any such method not present till now, except DNA.
yes, they will not. You have to just you cannot directly inject mRNA, and the mRNA will be degraded by the bacterial endonucleases. Okay. mRNA you cannot inject into the cell, and then you cannot expect it to express the protein. Okay. So you have to clone the DNA into a in a plasmid, and then you can express it. So that is cDNA. cDNA will basically not have any intron sequence. So that is the reason. Okay. Now, so what happens is, so how is this cDNA synthesized? So here they are asking you about how you can synthesize a cDNA. So what is done is like you have the mRNA sequence. Okay. Now this mRNA will have a poly A tail. Okay. So what is done? You take a primer. You take a primer having this is a poly T primer. Now, if you use a DNA primer having this poly T sequence, this DNA primer will go and bind to your mRNA sequence. Now, what will happen? The DNA polymerase, you can use the DNA polymerase, and a, a complementary DNA sequence will be synthesized, right? You will get a complementary DNA sequence. So this is your RNA, and here you will get your DNA. Okay, so this is what you are getting is a RNA DNA hybrid. So you are getting an RNA DNA hybrid. Now you want to degrade this RNA. So if you want to degrade this RNA, you have to use. So how
RNA.
you synthesize DNA? Yes. So how you synthesize? So I said it wrongly actually. So you will use a poly T primer, and you will not use DNA polymerase. You will use reverse transcriptase enzyme. So if you have to synthesize a DNA from an RNA, you need a reverse transcriptase enzyme, right? Just give me a second. Uh, so did you understand what I was explaining? So first you have the poly mRNA having the poly A tail. You use a primer having this oligo T sequence. Now this DNA primer will go and bind to your RNA having this poly A tail, and then you can add reverse transcriptase. So once you add reverse transcriptase, the complementary DNA sequence will be synthesized, and then for degrading this RNA. So if you want to degrade RNA from a RNA DNA hybrid, then you need the RNase H. So once you add RNase H, this RNA fragment will get degraded, and then DNA polymerase is used for synthesizing a complementary DNA sequence. So this is how you get the cDNA. Okay, so this is how you get a cDNA. So that is what is explained here. So during the Gober-Hoffman method approach for cDNA synthesis after first strand synthesis using oligo-DT primer in the presence of dash and DNTPs. So what do
Sir,
you need here?
after
Yes.
adding after means in the last step when the
Hmm.
RNA is degraded with
Hmm.
RNase, then to make the other strand means the left out strand complementary,
Hmm.
then we will add the DNA polymerase so that the other strand is formed.
Yes, other strand will be formed. So you have the remaining DNA sequence, and using this DNA sequence as a template, your DNA polymerase can synthesize the complementary DNA sequence, right? So it will use the 3' OH end, and it will synthesize the complementary DNA sequence, isn't it? The DNA polymerase. So now you read the question carefully. So during this approach for cDNA synthesis after the first strand synthesis using oligo-DT primer in the presence of dash and DNTPs. So what do you require other than nucleotide triphosphates? What is the enzyme that you will be requiring? So you will, so for the first for the synthesis of
Reverse
the first strand,
transcriptase.
yes, tell me.
Reverse
Primer,
trans
you will yes, reverse transcriptase. You will require primer, DNTPs, and reverse transcriptase. Why do you need to require reverse transcriptase? Because you are synthesizing a DNA from an RNA. Okay. So of AS reverse transcriptase. Now, in the next step, what is done? The DNA RNA hybrid. So after the synthesis of the first strand, you will get a DNA RNA hybrid. Now, this DNA RNA hybrid is treated with dash to produce nicks at multiple sites. Okay.
RNase.
Yes, RNA is H. RNA is H. Okay. RNA is H. And then dash is used to perform DNA synthesis using multiple fragments of RNA as primer to synthesis new DNA strands. Okay, so
Both.
what actually RNA is? So that is DNA polymerase. So your correct answer is option A. Is it clear now?
Option C, sir.
No, no, not option A. Option C.
Option D.
No.
Option D. Option D. Option D. DNA
Yes, it
polymerase.
is option D. It is option D. Yeah, sorry. A reverse transcriptase, B RNase H, and C is DNA polymerase. Okay, is the question clear now?
Yes, sir.
Okay. This was also explained in your in the lecture, video lectures also. So you see this figure. Is it clear now? So you have an mRNA with a poly A tail. Then you add an oligo DT primer. So this oligo DT primer, it will have this TTTT tail, and along with it will also have a complementary DNA sequence. So that this primer can go and bind to this mRNA. And then you add reverse transcriptase and the four types of dinucleotide triphosphate. What are those? They are DN, DTTP, DATP, DGTP, DCTP. Okay, we will add these, and then this reverse transcriptase will synthesize the DNA. And the next in the next step you add RNase H and DNA polymerase. So this RNase H will fragment this RNA primer, this RNA mRNA into, it will create different fragments of this mRNA, and then your DNA polymerase can come. DNA polymerase one is actually used. It has this 5 prime to 3 prime uh, polymerase activity, 3 prime to 5 prime exonuclease activity. So using all this activity, it can synthesize the remaining DNA. Okay, so now the last, no, two more concepts are remaining. One is this SDS page. So have you ever prepared an SDS page in your practical class or in your laboratory?
No, sir.
So agarose, so agarose gel electrophoresis is used for what? For separating your DNA fragments, right? So if you want to separate your DNA fragments, you use agarose gel electrophoresis. But for separating proteins, what do you need?
Southern.
Polyacrylamide. You need to prepare a gel having
So we can do this by southern blotting also. So I think there is network issue. Hello.
yes, yes. Is my screen visible? So why
Yeah.
do you need polyacrylamide?
We can do, sir. We can do this by sorry, southern
Why
blotting
can't you
also.
separate proteins using agarose? No, now we are not talking about blotting. We are talking about electrophoresis technique.
Okay, sir.
So if you have, uh, so if you lyse a cell, for example, you have lysed a cell, and in your lysate you will have many different types of proteins. So you want to separate the proteins based on their molecular weight. So you have to run a SDS page. So for DNA, you run an agarose gel electrophoresis. For proteins, what do you need? You need to run an SDS page. So why do you need to prepare polyacrylamide gel for proteins? Why can't you prepare agarose gel for proteins?
Uh,
So because if you, so this is actually has something to deal with the pore size. So if you look at the pore size of agarose, agarose has a very large pore size. The pore size is while am I audible? Okay, so remember that agarose gel have larger pore size. And why do you require larger pore size? While polyacrylamide gel have.
to separate
Polyacrylamide.
the DNA fragments means larger the size will remain and will remain the farthest, and the smaller size will move through the pore and go faster than the
So,
larger
so,
one.
so if you, for example, if you add DNA, if you load your DNA in a polyacrylamide gel, then will it not be separated? Can this polyacrylamide gel resolve DNA fragments?
But sir, here as
So why do you require large pore size for DNA while small pore size is required for protein? Why do you
due to the
so,
size of
so
DNA,
yes, why? Because what is the size of DNA? Because of the size of DNA. Yes, you are correct. But can you be more specific? Like uh, DNA. So if you know the
it's
average
10
molecule,
to the power
yes,
something 8 or like this base pairs, and protein are only of 50 or 100 amino acids.
yes, 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 your model is correct. So if you look at the molecular weight of a single nucleotide, so the molecular weight of a single nucleotide is around 660 Dalton. It is around 660 Dalton. While if you what is the molecular average molecular weight of a single amino acid?
128 something.
Yes, it is 110 Daltons. Okay, so the average it is not the absolute. It is the average molecular weight of one amino acid. It is around 110 Dalton. Okay, so for example, if you have a protein of around say 300 amino acids, and if you have a DNA, if you have a DNA of 300 base pairs, you can calculate the molecular weight now. So if your one base pair is of 660 Dalton, then so one nucleotide is of 660 Dalton and the DNA is double stranded. So this will be double of that. And then that multiplied by 300 will be, it will give you a very high molecular weight compared to proteins, which will, if it is having 110 Dalton, then its molecular weight will be around what? It will be around 300 into 110 Dalton. So yes, because of the molecular size only that proteins have so if you compare the protein and nucleotide having the same number of amino acids and same number of nucleotide bases, then the molecular weight uh, of a of a DNA will be more than that of an amino acid. So for DNA, you require gel having larger pore size compared to proteins because they're having smaller molecular or smaller molecular weight compared to the DNA. Okay, so now what is an SDS page? So SDS, the term SDS stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. and it is a widely used technique for separating mixture of proteins based on their size, only based on their size, not on the basis of their shape or charge. So protein will have what? Proteins have size, it will have a particular molecular weight, it will have a particular shape. Proteins have different conformations, right? Secondary structure, tertiary structure, and also it will have a charge. So if you want to separate the proteins only on the basis of their size, then the what is done is you need to run a SDS page. So what is SDS? It is an anionic detergent, and it is an anionic detergent means what? It will have a negative charge because of this sulfate group. Because of this sulfate group, it will have a negative charge, and this negative charge is used to produce an even charge across the length of the protein that have been linearized. So once you have the protein, and if you treat it with SDS, your protein is will be folded, right? But once you treat your protein with SDS, the SDS, what it does, it linearizes your protein. So it converts the tertiary structure of a protein into a linear structure. Now, once it gets converted into linear structure, this SDS adds negative charge to these proteins. So it uniformly coats or adds negative charge to the linear chain of the protein. So now, when you linearize the protein and you have added a negative charge uniformly along the length of the protein, then the only thing that is left is the size. So now the protein molecule will get separated only on the basis of of their size and not on the basis of their charge or shape. Okay, so this SDS is doing two things. First, it is linearizing the protein. So the effect of shape on the mobility of the protein is removed. Okay. So there is no effect of shape and there is no effect of charge. Now the protein will get separated only on the basis of their molecular weight. Okay, because it adds a uniform negative charge to the protein and it also linearizes the protein. So there is the charge to mass ratio. This becomes equal. So by first loading them into a gel made of polyacrylamide and then applying an electric field to the gel, the SDS coated proteins are separated. Okay, so the electric field acts as a driving force, drawing the SDS coated protein. So once you add the negative charge, uniformly negative, this protein is coated with uniform negative charge. So what will happen? So when you apply the electric field, these will move to the positively charged electrode. Okay, so it will move towards the positively charged electrode. And in order to identify proteins by size, the protein standards of a known size are loaded along with the sample and run under the same condition. So you have a ladder. So the ladder has proteins of different molecular weight. So alongside the sample, you run a protein ladder, and then comparing the ladder with your protein sample, you can estimate that what is the size of the protein or whether the protein that you have expressed is it having the required molecular weight or whether your protein has expressed. So all these things you can analyze. Okay, so what are the components of an SDS page? So the very first component is an SDS. So as I was explaining, SDS is an anionic detergent. So it is an anionic detergent and it is composed of a hydrophilic group with a net negative charge and a long hydrophobic chain. So this is the structure of sodium dodecyl sulfate. So it has an hydrophilic head. Hydrophilic head, why? Because of this sulfate group, and there is a hydrophobic chain. With this hydrophobic chain is CH2, CH2, CH2. This hydrocarbon chain is present, and this hydrocarbon chain blankets proteins in proportion to their mass at a rate of 1.4 gram of SDS per gram of protein. So different proteins are, so what will happen? It will give a uniform M by Z relative charge to mass ratio becomes equal for all the proteins. So now the proteins will get only separated on the basis of their mass and not on the basis of charge. Okay, this provides the protein with the driving force necessary for size-driven separation in an electric field. Okay, so polyacrylamide is a polymer. So as agarose is also a polymer, here polyacrylamide is a polymer that is formed from two components. It is formed from acrylamide and bisacrylamide. Okay, so acrylamide plus bisacrylamide together. So bisacrylamide is used for forming cross links between two polymer chains. Okay, so the more the concentrated, so you can prepare different concentration of SDSP gels. You can prepare 12% SDSP, 15% SDS gel, 18%. So depending on how much acrylamide you add, uh, that will determine what will be the porosity of the gel, or how, or whether your gel pore size of the gel will be small or whether it will be large. That will be dependent on the what is the percentage of SDS gel that you are preparing. So then there is a loading buffer. Loading buffer has three components. It has SDS and it has different reducing agents like beta mercaptoethanol, or it can have BTT. So all these, what it does, they are reducing agents. Why? What does a reducing agent do? It will reduce the SS disulfide bonds into SH bond. So it will, if there are any quaternary structure in a protein, that will also be removed. It will become a linear protein. Okay, and glycerol, why is glycerol present? Glycerol is present so that to add weight to your sample. So when you load the protein into the well, you would not want the protein uh, uh, to float above, or to get to be floating. So it adds weight so that your sample it gets properly loaded into the well, and then bromophenol glue is used as a tracking die. So you can analyze how far your gel has run, and you can decide whether you have to stop the gel or you have to run the gel more. Okay, so now answer this question. So polyacrylamide gel consists of chains of acrylamide monomers. So these are acrylamide monomers, and they're cross-linked with bisacrylamide. Bisacrylamide means it has bis means two, so it has two units of acrylamide connected by a methylene bridge. The polymerization of, so actually what happens if you have so when you prepare a polyacrylamide gel, a chemical reaction takes place. So it is unlike agarose gel. Agarose gel gets solidified on the basis of the temperature. So once the temperature comes down, the agarose gel solidifies. But when you prepare a polyacrylamide gel, then a chemical reaction takes place. So it is a free radical catalyzed reaction, and this reaction is initiated by the quaternary amine dash, which induces free radical formation from dash. So you have to remember that where there are what are the various components of when you prepare an SDS page. So what are the different components that you require? First, you require water, then you require acrylamide, then trace. Okay. Uh, then the fourth component that is required is SDS, fifth APS, and sixth is TIMET. So these are the six components that are required. And there is also, there are two different trace buffers that are required. One is having pH 6.8 and one is having pH 8.8. Okay. So what happens? So this is a free radical reaction, and the free radical is provided by ammonium persulfate. So ammonium persulfate is the chemical reagent which provides the free radical. Okay. And this free radical reaction is catalyzed by TIMET. So TIMET is the catalyst which catalyzes this free radical reaction. So TIMET is the quaternary amine. So A is TIMET and B is ammonium persulfate. Correct answer is option A. Okay. So this is actually explained very nicely in your lecture slide. So I did not make any other extra slide. So you can see over here also. So these are your acrylamide monomers. And then this acrylamide monomer. So you take basically, if you have to prepare a 30% acrylamide uh, in lab, then you take 29 grams of acrylamide and 1 gram of bisacrylamide. And then what happens is once you add this TIMET and ammonium persulfate, so ammonium persulfate generates your free radical, and this TIMET, the reaction initiation is done by this TIMET. Okay. So ammonium persulfate in the presence of TIMET forms oxygen free radicals and induces the polymerization of acrylamide monomer to form a linear polymer. So these linear monomers are interconnected by cross-linking with the bisacrylamide. So, so, so you can go through this. So it has explained it is explained very nicely. So this ammonium persulfide, remember that it generates the free radical, and the TIMET is the one which catalyzes the reaction, or it initiates the reaction. So if you do not add the timid, your gel will never solidify. You have to add timid. Okay, so what is the purpose of stacking gel in SDS space? So when you prepare an SDS space, there are two gels. One is the stacking gel and another is the separating gel. Okay, so the separating gel is present at the bottom and the stacking gel is present at the top. Okay, so why do you require two two components in an SDS gel? So this is your separating gel and this is your resolving gel and this is your stacking gel.
So option is it option C to concentrate the proteins
To
into.
concentrate the proteins into a narrow band. Yes, you are correct. Answer is option A. And how is it done? So in stacking gel, what is the pH that is used? What is the pH of tris that is used? And what is the pH of tris that is used in separating gel? In separating gel, the pH that is used is 8.8. Okay. And in stacking gel, the tris has a pH of 6.8. So what happens now? Your running buffer and has glycine also. Okay. So what will happen? The PI of glycine is around it is around 6.5. So in the stacking gel, since the pH is almost equal to PI, the glycine has a neutral charge. Glycine has a neutral charge, while in case of in the separating gel, because the pH is more than PI, the glycine will have a it will have a negative charge because the
So
pH
why
is greater
we
than
are
PI.
taking glycine?
Glycine, because glycine is a component which is present in your running buffer, SDS running buffer. So what happens when you run the gel, your protein. So what happens in the stacking gel? In the stacking gel, protein gets sandwiched between the glycine and the your protein gets sandwiched between the glycine and the chloride ions. So what happens is because this glycine has a neutral charge, it migrates slower. It migrates slower compared to your protein which is having negative charge. Your protein you have added SDS. You have added SDS to your protein. So when you add the loading buffer has SDS. So because the loading has SDS, the protein gets negatively charged. So your protein migrates faster than the glycine. So glycine is present behind. This glycine molecule is present behind the uh, protein, and the NaCl and the chloride ions migrate ahead of the protein. So what happens in the stacking gel? Your protein gets sandwiched between the chloride and the glycine molecule. So this helps in forming a very narrow band. So how is the protein concentrated in a very narrow band? So because of the presence of this pH, and the protein gets concentrated over here. And once now, when the protein moves into the separating gel, what happens in the separating gel? Now the glycine will have negative charge. So what happens? The glycine now starts moving ahead of the glycine will start moving ahead of the protein. So if this is a protein molecule, the glycine and the chloride molecules will move ahead of the protein, and your protein will get separated on the basis of your molecular weight. Okay, is it clear? They become hodgepodge.
Little bit.
What is the confusion? So you have to understand the concept of pH and PI. Do you understand the concept of pH and PI? So
Yes.
all proteins have a particular PI, right? So if you keep the protein in a buffer which is having a pH less than the PI, then what will happen?
Acidic.
And if you keep your protein in a pH which is equal to the PI of the protein, then what will happen? And
Means
if you
bitter
keep
ion
your
will
protein in a
form.
pH which is greater than the pi of the protein, then what will happen? Tell me first.
pH
What
less
will be
than
the charge
pi.
of the protein? Yes.
pH less than pi. Here the charge will be acidic, means
Yeah.
um,
Acidic means
negatively charged.
no, positively charged. It will be positively charged.
Positively
Okay.
charged, and in a
And
neutral
here it
case
will
it
be
will be
zero.
act,
It will be neutral,
and
right?
the
And
lower
here
one
it
will
will
be
be,
negative charge.
yeah, it will be negatively charged. Okay. So now you tell me if glycine has a pi of 6.5. Okay. So what will happen to glycine at pH 6.8 and pH 8.8? What will be the charge on glycine?
At pH
At
6.8
pH
it will
6.8,
it will
it
be
will be
negatively
more
charged.
or less yeah, but it will be more
More
or
or
less
less
it will be neutral. Means most of the molecules of glycine
neutral.
will be neutral. Okay, but highly negative. All the molecules will be negatively charged at pH 8.8. Right, right. Is it? Did you understand this point?
Yes, sir. Yes, sir.
Okay. So now if your glycine molecules are having negative charge at pH 8.8, so your protein is having negative charge. Your protein has a negative charge. Your glycine also has negative charge. So glycine is present in your SDS running buffer. Okay. Your
Repulsion will
SDS
be there.
running buffer have trace SDS and glycine. Okay. And it has also chloride radicals are also present. These are also negatively charged because it is tris HCl. So tris HCl will have chloride ions, right? So now what happens? This the in the separating gel on the or in the resolving gel, your chloride and glycine will move ahead of your protein. Okay, because they are having lower molecular weight, they will move ahead of your protein. While in the stacking gel, your protein gets sandwiched. Your protein gets sandwiched between the chloride ions and the this is your protein. This it gets sandwiched between the chloride and the uh, glycine molecule. Okay. And this helps in forming a very crisp band in the separating or in the stacking gel. Okay, so this is explained here. If you read this paragraph, you will understand. So the problem is taken care once the sample is run through the stacking gel. Yes, you tell me now if you have any doubt.
No, sir.
Okay. Okay, so now we'll move to the next question then. Okay, you read this paragraph nicely, will understand. Okay, so this is what I was explaining. The same thing is written over here as well.
Okay, sir.
So now electrophoretic mobility is directly proportional to the dash and inversely proportional to the dash of the molecule. This is this is very easy question,
Charge
right? Mobility. Tell me the answer.
inversely proportional to the size and shape, and directly proportional to the charge.
The charge. Yes. Yeah, this is a very obvious question. The more the charge, the higher will be the electrophoretic mobility, and the lesser the molecular weight, the higher will be the mobility, or your mobility will be directly proportional to a charge, but inversely proportional to the mass. Okay. So this is an easy question. So here also you can see same thing is written. So if you are the higher the charge, the higher will be the mobility, but it will also depend on the shape. It will also depend on the mass, size. So the size, or if the higher the size, the lesser will be the mobility. Okay. Now last concept for today is Western blotting. So Western blotting is used for identifying proteins or analyzing the expression of proteins or what is the abundance of a protein. If you want to analyze, you will do Western blotting. So Western blotting is an analytical technique used to detect specific proteins in a given sample of tissue, homogenate or extract. So you have a tissue or you have a bacterial cell or you can have a mammalian cell. You lyse the mammalian cell or bacterial cell. You will have a lysate, and your lysate will have thousands of protein. Now you want to analyze, identify whether that particular present, or whether that particular protein is present in your lysate, or what is the expression of that particular protein. Then what you have to do, you have to do a Western blotting. So it is used to identify the presence of specific proteins in electrophoretically separated samples. So similar to northern blot and southern blot, here also what you have to do, you have to separate the proteins by doing a running an electrophoretic gel. So for DNA or RNA, we are running an agarose gel electrophoresis. For protein, you have to run a SDS-based gel electrophoresis. Now, what will happen? First, so when you run an SDS-based gel electrophoresis, your proteins will get separated on the basis of the molecular weight. Now, then from the gel, you have to transfer the protein from the gel onto the membrane, onto the nylon membrane. And then again, you have to do a hybridization. So in hybridization, what you will use? So now you will use a probe. So what is the probe in case of Western blotting? It is an antibody. Okay, so because the antibody will identify a particular epitope on your protein, you will hybridize it with a antibody probe. And then this anti then so this is your primary antibody. Then you have to use a secondary antibody. So your secondary antibody will be tagged to a fluorophore, or it can be tagged to a enzyme like radish peroxidase enzyme, alkaline phosphatase enzyme. So when you add a substrate, then it will convert the substrate into a colored product. And then by quantifying the color, or by quantifying the fluorescence, you can calculate the abundance of the protein in your lysate. Okay, so this is the overall concept about Western blotting. So these also will have 5 steps. First, you need to isolate the protein. Then you have to run an SDS based gel. Then you have to transfer the protein from the gel onto the membrane. Then you need to do hybridization of the protein with a primary antibody. Then you have to do with a secondary antibody, and then you have to visualize it. Okay. So now, what is the purpose of secondary antibody in Western blotting? So why do you need a secondary antibody? So it increases the sensitivity basically. Okay. Uh, so your primary, if you want to, uh, your sensitivity increases when you use secondary antibody, because uh, uh, once you use secondary antibody, your secondary antibody will go and bind to the FC region of your primary antibody, and you can be sure that this is the position um, of my protein, or my protein is present, my target protein is present. Okay. So to amplify the signal from the primary antibody. So this is the correct answer. So the signal gets amplified from the primary antibody. Answer is option B. So in two-step probing, membrane is first probed with primary antibody to recognize the protein of interest. So for this, you can either generate monoclonal or polyclonal antibody, and then the membrane is probed with, is washed with buffer, and reprobed with another antibody directed against the primary antibody. So the secondary antibody is coupled with an enzyme or a fluorescent dye. The wash membrane is incubated with an appropriate dilution, and use of two different antibody increases sensitivity as well as flexibility. Okay, it increases the sensitivity, so you are more assured that yes, this is where my protein is. So the sensitivity increases. Non-specific binding, or the problem of non-specific binding reduces further. You're using secondary antibody. Okay, so now during the process of Western blotting, 5% skim milk is the major component of what? So as we had seen that for northern blotting, you need salmon sperm DNA for blocking the membrane, or for removing the problem of non-specific binding. Here, in case of Western blotting, you need 5% skimmed milk. So this skimmed milk have casein. So because of which it will go and bind to the regions of the membrane, and it will not allow the antibody probe to bind anywhere on the membrane, and uh, and enhance its or it will and as a result, what will happen, the noise that you will get will be reduced. Okay, so the first step of immunoblotting is to cover the remaining areas of the membrane with a dilute solution of protein. This step is called blocking, and it is performed to block the membrane and reduce the non-specific binding of the antibody. So you want to reduce the non-specific binding of the antibody, so you add this skimmed milk. You can add skimmed milk, you can add BSA. So all these are used, or you can add a non-fat dry milk. So this step is usually done for 1 hour to overnight on a shaker. So the membrane is washed with distilled water to remove the non-specific binding, and at the same time, before adding your antibody, before adding the primary antibody, the membrane blocking has to be done by adding 5% 5 fat BSA or skim milk. Okay, so that was all for today. Today's lecture. I hope these concepts are very clear, and if you have any more doubts, we can take up again in the next class. I will end today's session now. Thank you for your patient listening. Bye.